Good afternoon, everyone. I'm Dr. Rick Barron, the Executive Vice Chancellor for Health Affairs at MU and the Executive Director of NextGen Precision Health Initiative. I want to welcome you to the sixth session of our NextGen Precision Health Discovery Series. It's amazing to me that we have completed a half a year of this series, and I'm glad many of you have either attended live or watched the recordings posted on our NextGen website. These discussions are intended to keep us all updated on the NextGen initiative. We have asked our speakers to keep the discussion at a high general audience level rather than in a detailed specialty level. As you hear these wonderful talks, we would like you all to think about transdisciplinary research opportunities in precision health, where we can identify potential collaborative opportunities for our partners across the University of Missouri system and with our partners at Washington University in St. Louis. It's a pleasure to introduce our speaker today, Dr. Christina Gurnett. Dr. Gurnett is the Ernest and Jane G. Stein Professor of Neurology and the Director of the Division of Pediatric and Developmental Neurology at Washington University in St. Louis. Her clinical and research areas include pediatric neurologic disorders, epilepsy, and musculoskeletal disorders. And she is a graduate of the University of Iowa School of Medicine, where she also conducted her residency in pediatrics. She's completed 2 pediatric fellowships, one in neurology and another in epilepsy at St. Louis Children's Hospital, which, as you know, is affiliated with Washington University. She also serves as the Children's Hospital as its neurologist in chief. So it's great to have a fellow neurologist as our speaker today. Dr. Gurnett will discuss WashU's Institute of Clinical Translational Sciences Precision Health Function, which is a major component of the WashU NIH CTSA program. And as many of you know, University of Missouri is officially now a partner and a spoke of the WashU CTSA program. And at WashU, they call their program the Institute of Clinical and Translational Sciences. So today, Dr. Gurnett will talk about the efforts this institute uses to integrate genomic and electronic health record data and how it engages community and research participants through the return of research results to the patients. She will also discuss her own basic science research into gene variants and someday how this can help patients through translational research. So Dr. Gurnett, it's a pleasure to have you here with us today, and I'll turn it over to you.
Thank you so much for the kind introduction. I'm really glad to be here. Bill Powderly, who's the director of the ICTS, and we both made a visit to MU a couple years ago, met with Bill Fay and a number of other folks, and we're starting to build more collaborative efforts across our 2 campuses. And so what I want to do today is really talk about kind of 2 things. First, I'm going to give a little overview about the Institute of Clinical and Translational Sciences to give you an idea of the resources that are available to you. We'll talk a little bit more about the specific precision health function that I help co-direct at the ICTS, and then we'll segue a little bit more into some of the work that I do in my own research that helps us to know how to bring precision health to patients. So just to give you an overview of what is the ICTS, it's really a consortium of partners, including folks at University of Missouri Columbia, but we also engage our partners at St. Louis University, the University of Health Sciences and Pharmacy, and then our hospital systems at the BJC at Washington University. And so almost all of the resources, with few exceptions, that the ICTS supports are available to all of our partners and all of our faculty at MU as well. I'd first like to just really talk about what is translational medicine. And, you know, we think of research in the preclinical phase in T0, and then certainly translating to humans in T1 and you've heard the terms T3, T4, and T. And really the goal of translational medicine is to move between these categories, so to bring information more quickly into patients. And so that takes efforts to bring them out of the lab into humans for the first time, and then bringing them into clinical settings and then bringing them into the real world clinical setting outside of the ivory tower that we sometimes sit in at our academic institutions. And then how do we bring this to fruition by improving public health for all? So the ICTS does a lot of things, again, to try and improve the speed at which we bring improvements to community health. We do a lot of connecting. So we spend a lot of time talking to investigators about how to find the resources they need to do their work. We have a big component of translational science workforce efforts, all the way from the undergraduate level, all the way through faculty, but also extending to research coordinators, nursing support, all the things that we need to have a good workforce to do research. We work really to get the message out and to work with our communities and have a bi-directional exchange so that we know what they need and that we can be responsive to the needs of our communities. Not only, again, at our own institution, but how can we partner with our regional providers, including MU, to help do this. We work on disseminating information out into the real world. We do a lot of collaboration and team science building. Bringing more people to the table can make the projects even better. So we work a lot on trying to build more meaningful projects with the community engagement and with multidisciplinary scientists. And then I'll talk a little bit more about the funding that we offer. This is, again, always the fun part of my job, which is giving away money for pilot funds to be able to jumpstart your project and get it to the next level. So you should definitely visit the ICTS website where a lot of this information is also held. We've tried to divide resources into those that might be more pertinent to early stage investigators, and then also fine-tune links for clinical researchers, and again, for individuals who are working more in the population health space. We have those resources kind of pooled. Our largest funding program is our Clinical and Translational Research Funding Program, the CTRFP, which we're right in the process of. So we did have, I can't remember if it was 10 or 15 applications from MU this year. We just had our letter of intent that was due in June. We're sifting through those. And we'll be asking probably half of the 100 applications to go on to the final competition, and so they can submit a full application later in later in July, and we'll be reviewing these later this fall. These are one-year pilot awards for translational research efforts for up to $50,000 per year. We fund about the 20th percentile. They are reviewed in study sections, and we run these just like NIH review sessions, and we have asked a number of reviewers from MU to join us on this. And so it's a lot of fun because we get to meet people from multiple different institutions and get to know each other that way as well. The good news, we put a lot of money into these. We fund about $1 million per year, when we also can consider funding that comes from our BJC hospital system. But the return on investment is huge. These are, people who seek out these pilot awards tend to be very, very successful in generating the data, and that can bounce them into NIH level funding. And people who are engaged with the ICTS, we also have done studies, they tend to be successful with NIH grants. And we do avail them of some of the other services that I'll talk to. So if you engage yourself at some levels, there's multiple resources that you can have access to. The other funding program that I'm in charge of is our just-in-time core usage funding program. We have about 40 different core facilities at Washington University. There's one now at St. Louis University, and so if there are core facilities that you use at MU, we would love to include them as well in this to be accessible for use of these funds. We run this cycle once a month. It's a rolling call for applications. We review these very rapidly, and within the next month, if you're awarded, you'll have $5,000 worth of services to be used at any of these cores. these cores include very broad, genomics cores, biostatistics, imaging at our, high-tech Wookie imaging core, research data core. It's a really long list, and so you can kind of be creative with thinking about your project and where you can actually get preliminary data to build a new project. For some projects at our hospital system that would lead to improved patient care, we do fund those types of QI projects as well. We receive about 10 to 12 applications per month, and nearly all of them are funded. We want to get this money into people's hands quickly so that they can do the work that they want. And the return on investment for these projects is even greater. A lot of success comes out of these two funding programs. The other resources that we have at the ICTS, and we encourage a lot of community engagement. Some of these have not been used by MU investigators, but there's no reason that they couldn't. We have a community advisory board that can be convened. We did this during COVID when we were doing collecting patient samples, and we had questions about whether we should be doing sequencing on these patients who were consented under duress in an ER or in an ICU. And so we brought together a very diverse community advisory board to help us figure out whether we should be returning those results to families. What about if they're deceased? Should we be returning genomic data to the next of kin? So a lot of really great things come out of our community advisors. We have community engagement studios too, so individuals who are hoping to work with community members or who are working with community partners can have feedback in a roundtable discussion to know how to move that project along even further. And there are additional funding opportunities for community engaged research, including these PDSS awards, which are partnership development funding programs for about $10,000 to build capacity infrastructure with community partners. And then there is a special track for our CTRFP. Again, those applications, the letters of intent already came in last month, but we do have a separate review process for community engaged research because we do think this is so very important to what we do. I mentioned that, if you want to get engaged and want to start thinking about developing your research, we have great programs that are run by Betsy Keith, including an NIH mock study section that's held twice a year. These are typically about a month or two before the NIH review sessions happen. And so we love to see before the applications are due. And these can be very valuable to get feedback from 2 or 3, high-level investigators, experienced investigators who know how to kind of give you advice on career mentoring and your proposal. We've had MU applicants to the study section before. The reviews are all done online now, and we'll keep it virtual to make it easy for people there to access this. A more in-depth research program is our research forum program, where there's more of a roundtable discussion. You can invite your dream team of kind of investigators to help review your grant, and you can get advice from multiple people in the same hour and a half. And so these are hugely valuable, not only for new investigators and new projects, but a lot of us who've been involved have brought our own projects to these because we see such value in those programs. We do have a scientific editor who helps us also with our research and with a lot of the investigators who use the ICTS resources. Obviously, one editor can't do everything for everyone. And so your priority will go up for access to the editor if you have already engaged us either in the mock study section or the research forum program or one of our other just-in-time funding programs. I had mentioned Betsy Keith. She currently is our research navigator at the ICTS. So if you have a question about accessing any of the resources that I talk about in this talk, please reach out to myself or to her. We'll be happy to help direct you to the right core or the right paperwork to fill out in order to have access to what you need to do your research. I also mentioned the Clinical Research Training Center. They have also a nice website, and as I mentioned, they have a really nice range of funding opportunities all the way from the undergraduate level through postdoctoral and junior faculty programs, including the KL2 Career Development Awards. And these have been used in the past by MU faculty, and so if you want, we can certainly let you know who those people are, and you can have firsthand comments on how valuable these programs were to people's career. So I'm going to shift just a little bit to talk more specifically now about precision medicine resources. This again is just one part of the ICTS, but a part that I've been very involved with. We also have a website, so you can go find anything that I've talked about in even more depth on that website. As I already mentioned, some of what we do is really helping to build projects. We can identify in this space more specific content experts in precision medicine and precision health. We also run some outreach and community engagement types of efforts, including with the Venture Cafe. If you're not familiar with the Venture Cafe, it's a nonprofit that is actually an international nonprofit group, but we do have one here in St. Louis. Before the pandemic, we met in person over free beer. They meet once a week or every Thursday. And so we have content, and we invite lecturers to now participate with this. The Venture Cafe really is working to, with the entrepreneurial community, to get projects and things that happen in our space in the academics out into industry. And so it's a community program with lots of people with very diverse backgrounds that come together. They're all actually a lot of fun. The other thing that our precision medicine resource has worked on is how to access some of the really, really big genomic data sets. So some of you have probably heard of the UK Biobank, which is 500,000 people that were recruited in the United Kingdom and have corresponding in-depth health records, including brain scans, body scans, lots of omics data. And so there are ways to get access to the UK Biobank, and we can help advise people on how to do that. I think there could be some restrictions. A lot of the arrangements are such that we have one copy here for WashU investigators, but we're happy to help. If there's a lot of interest, we can let you know how we've done it here, and we'll be doing something similar now with the data that's coming out of the All of Us program, which I'll talk about in another slide. But there will be lots of large data sets, and I think it's really great to know that there is some expertise, and you don't have to have every investigator figure out how to do the paperwork behind some access for some of these very large data sets. Educational opportunities. I direct a precision medicine pathway for graduate students. We're doing a lot of those over Zoom, and so we could certainly invite MU graduate students to that. We have a Genomics in Medicine series that has been open, and we have had a lot of MU faculty join that, which, and then we'll be adding your NextGen Precision Health Seminar that you're attending today. We'll be adding that to our listserv, and happy to continue to provide access to these resources across our consortium. So one of the first things we did when we were leading this ICTS precision medicine effort 5 years ago was to try and find out what does the public actually know about precision medicine. It's kind of a term that means, as we've done this, means different things to a lot of people. So we really brought a diverse group of both rural and city folks with different racial backgrounds to kind of a focus group, so we could understand what are the needs and understanding of this term precision medicine. The good news was that virtually all participants had familiarity with genetic testing. It's not surprising. This is moving into cancer, children with disabilities. These are becoming more and more common in the clinic. but none of the participants were familiar with the term precision medicine. Of course, once we explained our idea of what the term meant, most people had a positive reaction. And then we started to talk more about returning results from research studies. And again, this fits with what we're finding on a national level too, is that people, if they know that we have their genetic data and it's meaningful for their health, most people say they would want to be informed. Our community advisory board that we held again for COVID patients where we were doing genomic studies, they also felt very strongly that even in those situations where we were bringing patients in and collecting their DNA when they were quite ill, they thought we should go back to those families and let them know if we had genetic data we needed to return. We just wrote the renewal for our ICTS grant, which was, it's a 5-year award. It'll be reviewed later, so we don't actually know if it's funded. But in the new reiteration, we changed course a little bit. Precision medicine has a little more narrow focus, and we really wanted to move more toward precision health and moving up the T0 to the T4 ladder to think more about health of populations. And we can start thinking about health of populations now because we have such robust electronic health record data and population data that's coming into our system. And our precision medicine function really works in this central white area where we really are centered around patients who are recruited for research studies. We're working with our mobile health research core, which is again open for just-in-time funding. So how do you do electronic consent? How do you collect data from wearables and mobile health technologies and include that into the data that you're collecting about patients? And then we are working a lot in this space where, again, participants are no longer happy with just being the participant. They want to actually be engaged in what we're doing. And so we feel it's really important to have bidirectional communication so that we're returning results back to families. Again, this is not what we've classically done. It costs a lot of money to do it. So I'll talk a little bit about the ideas and the ways that we're thinking about helping investigators return information to families. But I think this is going to be really important to keep the community engaged in the research process. Some of you may have heard of the All of Us research program. This is a national NIH-funded effort to recruit 1 million people across the United States. And they want diverse patient populations from rural and urban areas and all different ethnicities. They want it to reflect all of us. The problem with the All of Us program was that it's been funded, I think, around 4 to 5 years now, and the recruitment really stalled. They've been really dependent on face-to-face recruitment with participants, often in clinic settings, and then certainly with COVID, that pretty much diminished the ability to do face-to-face recruitment. And so we were asked and ended up obtaining some supplemental funding to have a pilot program that's called, they call it kind of the All of Us Light version, in which case we're doing virtual recruitment to the All of Us program and also connecting the patients up to their electronic health records so that we can donate that electronic health record data to the All of Us program. We've done this over the last 6 months, actually quite successfully. We used one of the ICTS resources, which is our Volunteer for Health program. We have 15,000 people enrolled in Volunteer for Health. And at relatively low cost, we sent out a message through MyChart to those 15,000 people. And with a single email, ended up with 500 people who enrolled in this study. They're also having to, again, donate their electronic health record data and send in a biospecimen. But I think this is a great model for how should we be doing research in the future, meeting patients where they are and not actually requiring them to come to us, and take advantage of some of the social media and the electronic health record integration into large-scale genomics and public health efforts. The other project that I'm very involved with, I'm the co-director for our Intellectual and Developmental Disabilities Research Center, which is one of 14 centers across the country. My co-director, John Constantino, wrote for supplemental, well, actually it was a grant through NCATS, which funds all of the CTSA programs, that we've been calling the Brain Gene Registry. And the goal here is to collect data from all 14 of these sites, not only, but on specific patients with particular gene variants or genes that have been mutated. And the goal is to collect data from all these health record, from electronic health record at each of the 14 institutions, centralize the health record data, integrate with it the genomic data, and really high depth phenotyping, including eye tracking software, lots of questionnaires, so that we can have deep phenotyping of these patients with disabilities along with their health records and their genomic data, and to centralize it to a resource that many people can access. So I think this, again, it kind of describes the very large scale types of efforts of sharing data across multiple sites that are going to be increasingly important to know how to do. And it sounds trivial, but there are a lot of steps at which this can fall apart. And these are really team science projects at their best. But I think the payoff will be good once we can figure out how to do this. The other thing the ICTS has been working on is coming up with an institutional genomic consent. And we really partnered with our institutional review board, who also saw that there was a lot of problems in the current practice where everyone's using different templated language. There's no standard. And so when you're trying to compare and share data across multiple different investigators at the university, you actually don't know. You have to read through the consent forms to figure out if it really allows data sharing. Do they allow genomic data or genome-wide sequencing data to be done? And so we have a standalone consent that we've developed, and we're really happy to share it with anyone. And I know that the NIH is also very interested in what is the standalone consent? What does it look like? I just got a call for information that NIH wants about this type of model. So we have a consent form that's been built. It really has three key things in it. It allows for data sharing, very broad data sharing. It allows for linkage to the electronic health record, and it does allow for some return of results. Right now, we've been using a 2-consent model, and so as investigators at WashU want to use our consent, they're typically having their own primary study consent, and then they're also signing this institutional consent, and we're keeping track of who has the institutional consent and which patients have been consented, so that when we start sharing data sets, we'll know that they've been consented appropriately. And the consent language that we use in our genomic consent is, if we discover genetic changes that are important to your health, would you like us to notify you about the results? And then we do extend that to, next of kin who might also be impacted by some genetic abnormality that we might find in a participant. And, return of results used to be somewhat problematic. People would, especially for genetic data, they were not sure they wanted this information to be returned. But, in all honesty, we've been doing this for a long time for other types of health data that have arisen during research efforts, including, and I've been involved in a number of imaging studies where we had to call patients when we found they had Chiari malformations, where their cerebellum protruded in, below the foramen magnum. And so we've had to have those difficult conversations with families where you find something you didn't expect to see. And doing this for genetic data is really very, very similar in the approach. Although obviously there's probably a lengthier list of things that we can identify through genetics research. So we've been basing a lot of our guidance on the National Academy of Sciences report from 2018. And with this model, they really take note about the value that the data has to the participants. And so when there is value for something that impacts their health, the return of results really should be considered as a matter of reciprocity and respect and trust, so that we can build that with the communities that we're serving. Obviously, reliability and validity of the findings are really crucial. You can lose trust very quickly if you return something that's not valid or reliable. And then certainly the autonomy. The participants should be able to decide if they want this result in return to them. And then communication and inclusion of diverse populations is something that we're going to continue to work on through the next 5 years. So secondary findings in genetic testing are those that are found unrelated to the primary purpose for the test. So if you find a BRCA1 variant, so the breast cancer gene, if you happen to find it when you're sequencing someone, but you know that there's something you could do to prevent that if you knew that someone had a pathogenic variant. And very commonly, there are also LDL receptor variants that are unrecognized, but should be treated early if an individual has that variant. So there are 59 clinically actionable disease genes that the American College of Medical Genetics lists as those in which if a pathogenic variant is discovered, it should be returned to a participant. And these genes include a lot of things like the genes that cause Marfan syndrome, long QT syndrome, things that have treatments. And so these are the genes that we typically would be looking at when we're doing a large-scale genomic or exome sequencing study. And I'll segue a little bit into some of the work that I do in my own lab, because obviously if the validity and the utility is really only apparent if you're very confident that the variant or the gene that you're talking about is pathogenic. And so we certainly come across lots of variants when we do sequencing. None of us have very large families, so it's often hard to know if that variant segregates with disease in a family. We can use to our advantage a lot of the really large genomic data sets that are already out there, including NOMAD, which has been collecting mostly healthy exome and genome data from about 250,000 people. And then disease mutation databases are also very, very helpful. And I'll talk a little bit about those. And all of us will be one of those large efforts where we're collecting data on millions of people, and so we'll know what is the frequency of a variant in the general population. The same with the UK Biobank, rich source. There's also a Million Veteran program that will have data again on mostly healthy individuals. We've tried to be inclusive in these data sets, but there are certainly populations that are not well represented. There is a new study of human health and heredity in Africa that's recruiting more diverse populations from Africa. And I do say that health equity in genetic testing is a huge problem. A lot of the data sets that have been sequenced for disease-specific sequencing have often been Caucasian populations. And so we really do need to make a huge effort to be inclusive in collecting genetic data on diverse populations. The other thing that I'm going to say here is a really big win is with data sharing. It wasn't that long ago that some of you probably will remember this story. So BRCA1 is one of the main inherited breast cancer genes. It was actually sequenced and identified and patented way back in 1994. And a company by the name of Myriad Genetics, which came out of the University of Utah and some academic partners, had patented BRCA1. And so for many years, a lot of us, even in research, we were, wondered whether we should be looking at this gene at all. It went all the way up to the Supreme Court, which decided in 2013. Some of you probably remember where you were when Elvis died or when various important things happened. I remember distinctly where I was when I heard about this Supreme Court decision, since many of us in genetics were really waiting to hear what was going to happen. They barred the patenting of the gene. It was a unanimous decision, 9-0. Even Clarence Thomas said Myriad didn't create anything. But unfortunately, what it still allowed was that Myriad held on to its large variant database. So even though other companies were now able to sequence BRCA1, everyone still sent all of their samples to Myriad because they had such a rich data set so that they could identify which variants were truly pathogenic and which were benign. Of course, there was a lot of public outcry about this. A lot of women had their genes sequenced and knew that they had a BRCA1 variant, and they wanted, whether it was pathogenic or benign, shared with the rest of the world so that people could make the right decisions for these really important health changes. And so a lot of women donated their data into large collaborative projects. And so this was really sharing their clinical reports so that other people could benefit from what Myriad had gained over years and years and years. And this really is truly a success story. Data sharing is now not only common, but it's really expected amongst the genomic companies that do any kind of germline sequencing. And you can go to the ClinGen resource and look at ClinVar, and it will list all the different companies that are doing sequencing and how many of those have actually submitted the variants they've identified, and not just the variants, but they've also submitted their interpretation. So if they've called a variant pathogenic and what are the reasons why they're calling it pathogenic, all that information is now in the ClinVar program. And so this is a huge, huge success for variant interpretation. You can also, a lot of effort has been made into can you predict whether a variant will be damaging based on the amino acid change or the level of conservation across multiple different species. And these are improving, but I think overall they're not perfect, and they really don't add a lot of utility for variant interpretation for specific patients with mutations. So then we're left with, how do you functionally test a different variant in a patient with a variant of uncertain significance? And how do you do that in a timely manner? So I've done a lot of research. I've made a lot of mice with single mutations, and spent, a lot of graduate students spend 4 or 5 years studying one particular mutation. And so it's too time consuming to make individual variants in model organisms, although that can be enormously fruitful for identifying the pathogenicity and the other disease-causing mechanisms. You can start thinking about using patient-derived iPS cells, but those actually are hugely expensive. I think it's close to $2,000 or $3,000 per line to develop an iPS from a patient. And so if you're going to do this one at a time across all 4 billion bases, this is going to take us forever if we're doing it one at a time. And so my lab and a number of other labs across the country have been really working to push the technology forward to do something that's called deep mutational scanning. This goes across lots of different names: saturation mutagenesis, massively parallel reporter assays. And the goal is to empirically quantify the functional effect of every possible variant in a gene. And often we're using a combination of single cell assays and next generation sequencing to really harness new technologies where we can, in a single assay, test thousands of variants. And this is going to have a huge impact, not only on patient care, because when we have a patient variant that comes back as a variant of uncertain significance, we can open that book and look at the table and find out quantitatively how that variant behaves compared to every other variant in that gene. And I'm also really particularly excited about what this is going to tell us about the basic biology of proteins, because to this date, very few proteins have ever been analyzed where you know and can quantify what's happening at every particular amino acid. So I think it's going to be fun because it's delivering lots of information that's going to be very clinically useful. So one of the best papers that's been published in the last couple of years came out of Jay Shenduri's group at the University of Washington, who did deep mutational scanning for BRCA1, the breast cancer gene, in a cell line that's called a HAP1 cell. These are haploid cells where they use CRISPRs to introduce variants into the endogenous gene. And HAP1 being a haploid cell, you can imagine there's some genes that are just absolutely essential for growth. And if you don't have that gene, or if you have a variant that's damaging that gene, the cells won't live. And so you can use growth as your assay and sequence at 2 different time points. after many weeks or in culture, many of the cells will die that have had damaging variants. And so doing this, he could come up with a quantitative score based on the number of reads that are present in a population of cells at 2 different time points. And clearly the nonsense variants in this gene were dropping out. And so he could build really every single nucleotide change, what would happen and what did happen in a growth assay, which very, very highly predicted which variants would be pathogenic and which were benign. So this is very powerful technology. My lab has been doing this. We've been working on trying to build different ways of making libraries. And I guess I'm just a lazy scientist who likes to get all the data in one single experiment. And this is really what we did in this experiment, where we were mapping out the active domain in the beta-lactamase gene, which confers ampicillin resistance. And in bacteria, we were able to introduce a library that we synthesized in my lab with every possible nucleotide change, and then grew in the presence and absence of ampicillin, and could see differential effects on the growth of these bacteria in the presence and absence of ampicillin. And in one experiment, we mapped out which of the amino acids were most essential for this function. We're now applying this technology to a neurologic disease. So Dr. Barron, always happy to come back to my home in neurology. We're applying this to a disorder that's called GLUT1 deficiency syndrome. And this was named after Daryl DeVivo, who actually was practicing here at Washington University many years before I came here, but still really leads this effort. He identified this gene, which is a glucose transporter in endothelial cells and astrocytes in the brain. And if you can imagine, if you don't quite have enough of this transporter, your brain actually doesn't grow. And so children who have this disorder end up with acquired microcephaly. They have severe seizures, movement disorders, and intellectual and developmental disabilities. In its severest form, it's quite rare, 1 in 24,000. You can diagnose this quite easily if you can do a spinal tap, which is not always that easy to do in children, by having low glucose in the CSF. More recently, though, certainly we've identified lots of variants that have come up in sequencing in this gene, SLC2A1. Many of them are de novo variants that aren't present in the parents. Most of them are single nucleotide variants or missense variants in this gene. There is a treatment, so we use the ketogenic diet for intractable epilepsy, but in this case, the use of ketones can bypass this defect in glucose transporter and improve some of the symptoms for these patients. What we've learned in the last couple of years, though, that there are a lot of atypical cases that are due to variants in this SLC2A1 gene, including absence epilepsy in fairly high functioning or normally functioning children, writer's cramp, there are episodic ataxias, and cerebral palsy. And so we find these variants of uncertain significance quite frequently now. And so this is unpublished data from my lab, but I have a postdoc, Mojgan, who has been developing similar methods using a growth assay, again, because HAP1 cells require this glucose transporter to grow. So when we sequence pools of cells at 2 different times, where we've introduced via CRISPR with every cell getting a different variant, we pool the cells and we look at the ratio of the reads of cells with the variants at 2 different time points. And by doing this, we've now taken some kind of gold standard positive controls, and we're able to clearly identify that these blue these blue bars here show that the variants are damaging. We had one patient that was unknown who had milder disease, and not surprisingly, her variant was mildly affecting the function of the protein and not within the normal range for what we think are most of these benign variants. And so we think this is going to be a really great tool to now query every possible variant in the gene We've got just a little bit of preliminary data where we've taken a library of molecules across exon 10. So I think there were 240 different variants that we tested. We introduced them by CRISPR and now can clearly define the red variants here that are damaging, and most of those being ones that are already known to be pathogenic, or certainly going to be informative for patients who end up with variants in those genes. Last couple slides here. All the data that we get needs to be integrated back into the variant interpretation framework, so the ClinVar and the ClinGen program. And so we've been working with folks at the McDonald Genome Institute on developing ways to present the data so that you can see the ranges of quantitative abnormalities and where the particular variant is that you've identified in a patient and that's been tested in our assays. And again, when this is done across multiple different assays, you can combine data from different publications, different investigators to give even more confidence in interpreting variants. So once you know that a variant's pathogenic with some of the work we do, how do you actually return research results to a participant? And we were just asking, at Washington University, how many people are doing this? How many people are giving information back to a research participant? And so we asked our IRB, and they reviewed 7,000 different IRB protocols, and 700 of those were doing genomics research. And then we specifically asked, are you returning results to participants? And 44 people said they were. When we asked who was actually doing it, though, a lot of us are kind of sitting in this, waiting zone, trying to figure out how to do this right. And really the number of people actually returning results is pretty small. And so we've got guidelines again on our website about how to do this. We're setting up some resources to have a kind of return of results clinic where we would have the samples sequenced in a CLIA lab, kind of vetted by a pathologist and a team of clinicians and geneticists with a genetic counselor who's involved in returning the results. And then we're trying to figure out, how do we do this? How do we sustain the ability to return results? Because it can be quite expensive for individual variants, but we think it's essential to build trust with our communities. It's going to require investigators to build the cost of doing this into their research. One of the proposed mechanisms that we're working with our Genome Center is to think about, can we actually include, a $10 surge charge on every patient who has, or any research participant who has exome sequencing or whole genome sequencing. And so we have kind of a pool of money to help subsidize the funding of returning results in that small, 1 to 5% of people who have a variant that you need to return to them. So with that, I'm very happy to give credit to the entire precision medicine team, including Laura Beirut and Megan Cooper, who've led a lot of this. We've had a number of very large committees, team science for how to return results, and then, of course, this is my own research group. So I'll be very happy to answer any questions.
Well, thanks, Dr. Gurnett. That's a great talk. I learned a lot. I guess some of the things that you and I have talked about before, that I'd like to ask you about now in public. Well, first of all, so these variables of uncertain significance that you then go on to determine if they're pathogenic, are these variables that are coming up through research genetic testing or through commercial genetic testing that you then put back in a report?
Yeah, that's a great question. I mean, it comes up in both. Through the ICTS, we're really focusing more on the research-based testing, but every day it comes up in clinical use too.
Well, that's right. And so you and I talked about this. So most of us are sending our genetic testing out to a number of really good, reliable clinical labs that do this, CLIA certified labs, and they can send you back the results that are either pathogenic or variables of uncertain significance. And so I guess you can take that information and then you can explore them on using research approaches like you're talking about.
Absolutely.
And so could you give us a little bit of the WashU experience? You all do have a CLIA certified lab, but I know in talking to you, you were telling me that still most of your genetic sendouts are done to the commercial labs. And it turns out that that wasn't that important to have a CLIA certified lab at WashU. And I know this has been a debate here, should we do it or not? And your input was really helpful for me on that. Can you tell the group what you told me?
Yeah, so we do, certainly our germline variants will, and exome sequencing for, children with disabilities, those we've been sending to a commercial company. I think, the companies have they have a couple things. They have really large data sets and databases. So when they're making calls on variants, when they have teams of 50 or 100 people who are used to doing it, the scale and the cost has always ended up being less than what we've been able to figure out if we were to do it internally. We've had a couple niche areas there. We've done some targeted clinical testing for cardiomyopathies and some other things that we have kept in-house. So they're, but for the most part, I think the companies have been.
Yeah, thanks for that.
Yep.
And that if that's the WashU experience, and I think that really should inform what we do here at MU. So thank you. Mary, are there other questions that you want to field for Dr. Gurnett?
Yes, there are. Thank you, Dr. Gurnett. And to everyone on the line still, please feel free to submit your questions via that Q&A tab. So the first one is, how can a person become involved with the All of Us research program?
Yeah, so there is a national effort. So if you type in All of Us, you should come to the national page. They are recruiting virtually. So if you wanted to volunteer, they will collect your information and consent you all virtually. They will send you a sample cup so you can spit your saliva and send it back in through the mail. We will, and then they will certainly be asking to try to get your electronic health record data. That's going to be a little bit more of a challenge for anybody who has most of their health at another institution. But I think at some point that will be, there may be ways of contributing that kind of data as well.
Thank you. Next, we have a compliment, really helpful talk. Thank you. And I'm curious what definition of precision health medicine you gave your study participants that led to that positive reaction. What about this definition for the consent forms you mentioned? Is the public's lack of familiarity with the term an obstacle to consent at all?
Right. I think what resonates most with me and with the public is, when you start talking about precision medicine, then you're thinking about how do you deliver drugs so you have fewest side effects and the most benefits for individual patients. And so I think then people start to realize that if you're improving what can be delivered specifically to them, fewer side effects, better outcomes, that resonates pretty well. But I think what people don't understand is how we're going to be integrating not only genomic data, but data from the electronic health record about them and about the learning healthcare system, when you can look across thousands or millions of people and identify key information that can also improve their health. So that's really why we moved into a more of a precision health program, because we really want to start thinking about the bigger picture and all of and not just things that happen in the healthcare system, but we have zip code data and the geospatial data so that we know what's happening in their communities, what are the lead levels in their communities that may impact all the different things that we can measure. But we will have to figure out how to integrate that environmental data with the electronic health record.
Thank you. Are there any genetic studies related to COVID for people with the disease, treatment methods, and outcomes?
Yeah, I don't think we have any active genetic projects. We did early on in the pandemic. I was very involved in in helping to set up a repository where we recruited about 500 patients with COVID. And we've got connected to their electronic health record, and that there is genomic data coming back from that. So if people are interested in accessing or looking at specific questions on a subset of 500 patients, we have that. But I don't know that there's any other active genetic studies for COVID.
Okay, thank you. Are there lab screening methods to examine effects of splice junction mutations or promoter mutations of potential disease genes of interest? And fantastic talk.
Yeah, when I think about how to do the functional genomics work that I do, I just need, I need to partner with really creative, excellent investigators who know that protein the best, because I think you can definitely imagine how there would be ways to look at splice and, I think even in our growth assays, we can look at those splice junction mutations and promoter mutations. But I'm always eager and happy to talk with investigators who have, who are very knowledgeable about their particular gene of interest and thinking about how do you develop a really great assay for a new gene.
So that's a great call to those on the phone or on the line to reach out to you. Then our final question here, and feel free if you want to add one more, but how much interaction or collaboration is there between NIH-funded CTSA or ICTS programs and sharing resources, cores, or data?
Yeah, I mean, that is definitely a goal. There's a reason there are 62 CTSA programs. They should be working as a network. We are very, definitely within our network with you and with the folks at SLU. All of those cores should be open to you at our institution through the ICTS. I think, cross CTSAs, it's not been as easy to have that kind of collaboration. There's a lot of effort in building informatics platforms that will allow sharing of data across multiple different resources and institutions. So definitely look forward to that. That's been a big investment.
Great, then I'll turn it over to you, Dr. Barron.
All right, yeah, well, thanks again. And I know Dr. Waitman here at MU and Dr. Payne at WashU are working together on trying to harness that power in the EMR to share information across our campuses and across the whole United States. So once again, thanks so much for that presentation. It's really a great guide for us to follow here at Mizzou and for our NextGen research system-wide. So I'm excited for the future of precision health, clinical trials, research, and I hope you all who joined us today share that goal too. Please join us again next month for our NextGen Discovery Series, when Dr. Christopher Contek will join us from Michigan State University. Dr. Contek was in town recently. He was invited by Dr. Turpin and others because he is the head of Michigan State's Institute for Quantitative Health Sciences and Engineering, which essentially is another precision health initiative. They're a few years ahead of us, and the lessons that they've learned over the last few years were really instructive for me, and I think you all will appreciate them as well. So his presentation will be at noon on August 19th, and until then, enjoy the rest of the day, and we will see you in mid-August. Thank you very much for joining.